Today's lecture, I'm going to introduce you to proteins, specifically structure, as well as different techniques that are used to study protein structure, which will be relevant when we do protein structure articles, as well as look into other functions of proteins in cells. So just to remind you, proteins perform most of the functions of cells, and in fact, if you look at dry mass of cells, it would be largely comprised of proteins. So proteins are extremely important to the function of cells. Proteins are comprised of amino acids, and amino acids have the important basic features. There's an amino group, carboxyl group, both of which are attached to a central carbon, a hydrogen group, as well as what's designated an R group, which is a different cluster of atoms depending on the different types of amino acids. Amino acids are linked together into longer chains called polypeptides, which you can see down here in this figure. And the amino acids are joined by covalent bonds called peptide bonds, which are shown here. And that's between the carboxyl group carbon, which is a carbonyl group, and the amino group. During formation of peptide bonds, water is lost to form that carbon-nitrogen bond. Proteins can be in the length of 10 amino acids to as many as thousands of amino acids linked together into polypeptide chains. Because of the huge versatility and types of amino acids, there's 20 different natural occurring amino acids. Many different sequences can be linked together and essentially create many different structures, which lead to many different types of functions. The different types of amino acids that are naturally occurring, some of which are shown here. There's polar amino acids, which ultimately are going to have charged groups in that R group, which essentially will allow them to interact with water. Other amino acids have polar uncharged groups that will allow them also to interact more with water, so it makes them more hydrophilic. And then we also have a group of amino acids that are considered nonpolar, and then some that are unique. So these 20 different natural occurring amino acids are divided into separate groups based on their properties that they might add to the amino acid in regards to their R group if they're polar or nonpolar. And the sequence of these different types of polar, nonpolar amino acids will contribute ultimately to how they fold based on their hydrophobic nature, interactions that can form, etc. within that polypeptide chain. Cysteine is one particular amino acid that actually has a sulfur group, and cysteine residues can form disulfide bonds in different regions of proteins. This is a type of covalent bond, so it's a strong bond that's found in proteins that are usually found outside of the cell. When proteins fold, keep in mind these polypeptide chains of proteins are extremely flexible, and they can fold in a number of ways. The folded state or native state that these proteins will ultimately finally fold into are stabilized by many different types of non-covalent interactions, which are shown here. Ionic bonds, hydrogen bonds, van der Waals forces, and ultimately many of these different types of non-covalent bonds are going to be required to completely stabilize a protein. And proteins do take on a final state that's its lowest energetic state, and usually is going to be the maximize those different types of non-covalent bonds that can form. Keep in mind these interactions may form between backbone components of the polypeptide chain, amino groups, and carboxyl groups. These other types of interactions can also be between R groups, which is once again shown here. So there's different ways in which these interactions can form through different parts of this long polypeptide chain. Another important force that contributes to protein folding are the hydrophobic and hydrophilic components of the polypeptide chain. If a protein is folding in an aqueous solution, the hydrophobic R groups will ultimately assemble into the core of the protein, and it will then, the hydrophilic amino acids will surround or be on the outside of the folded protein state. So you can see this in this particular diagram, the different polar versus non-polar side groups of the amino acids, and when that protein assembles into a final three-dimensional shape here, the hydrophobic residues will reside inside, protected away from the aqueous hydrophilic environment that it's surrounded in, and the hydrophilic amino acids will then be able to interact with the surrounding medium. This would be the reverse if it were a protein that would be instead folded in a hydrophobic medium such as the membrane. We would see the opposite effect. This gives you an example of hydrophobic versus hydrophilic amino acids and their arrangement when assembled into certain types of proteins based on their environment. This happens to be protein cytochrome C, and the green is showing you the hydrophilic amino acids, and in this diagram, the red is actually showing you the hydrophobic. You can see that the hydrophobic red amino acid side chains are more towards the middle of the protein, whereas the hydrophilic amino acid side chains are actually on the surface of the protein. Now, just to go through the folded state, final folded state of proteins, proteins can kind of be divided up into how they fold based on certain levels. These long flexible polypeptide chains essentially will fold into some final three-dimensional shape, which we'll talk about that process. The first level of protein folding is what's called primary folding, and primary folding is just the sequence of amino acids. The sequence will determine the structure. You change the order or sequence of a protein, essentially you change the way in which it can fold, the interactions in which it can form, and you change the shape of a protein. Because we have 20 different amino acids to work with, we can create very different sequences and essentially then be able to create very different shapes, which allows for many different functions. Ultimately, what allows us to choose, our cells to choose which order of amino acids to put together comes down to sequences of nucleotides in DNA, in genes. So protein coding genes will contain sequences of nucleotides, which are then interpreted into a coding sequence on how to build a sequence of amino acids. So our proteins are ultimately directed by sequences of DNA, and that is ultimately used to make all of our different proteins. Beyond the primary structure, a polypeptide chain, we also can talk about something called secondary protein elements. And secondary protein elements are common motifs such as helices, which is shown here, and also beta sheets, which we'll talk about in a minute. Helical structures in proteins are segments of the polypeptide chain that coil upon itself, which is shown in this diagram as well. These structures are stabilized in proteins by hydrogen bonds, and specifically there are hydrogen bonds between carbonyl groups and amino groups that are at different points in the polypeptide chain. And in fact, they're 4 peptides away. You can see that there are multiple hydrogen bonds that are essentially going to stabilize that helical shape of this particular helix. When the helical structure turns, there's about 3.6 residues per turn. There are what are called right-handed helices and also left-handed helices. And this is a very common structure in many different types of proteins. There isn't a specific type of sequence that's required. As long as there's certain flexible amino acids in a certain order, a lot of times you'll get the helical structure forming. If sometimes proteins will actually interact together through helical domains, and they can form structures even like coiled structures that link 2 dimers together by helices basically linking in together. Helices are also very common in proteins that are found in membranes, such as receptors or ion channels, as well as many other types of proteins. A second type of second element are beta sheets. And beta sheets are also structures that are stabilized by hydrogen bonds, and stabilized by hydrogen bonds that occur between backbone components again, carbonyl groups, amino groups. The difference between a beta sheet and a helix is that segments of polypeptide chains interact together. And these segments, these sections that lie next to one another, will actually form hydrogen bonds between them to stabilize this structure, which is also shown here. When these beta sheets form, it takes on a pleated shape, which is observed in all these diagrams that are shown here. Beta sheets can be what's called anti-parallel and also parallel. This has to do with the segments that are used in the beta sheet if the sequence is in the same direction. So an anti-parallel beta sheet, the sequence actually reverts back in opposite directions. So if this segment was 1 to 30 and the next segment is 50 to 80, you can see that this segment is going in this order, whereas this segment is going in the opposite order. That would create what's called an anti-parallel sheet. Still, hydrogen bonds are holding these strands together. Parallel sheets, the sequences run in the same direction. And additional looping above them allows for those sequences to run in that same direction. We see both parallel and anti-parallel sheets in different proteins. So beta sheets and helices are common. Do all proteins have helices? No. Do all proteins have beta sheets? No. Sometimes they can have both. Sometimes they can have one or the other, and some proteins may not have either. But once again, these two protein motifs are common because they both involve hydrogen bonds between backbone components that all polypeptide chains have, carbonyl groups and amino groups. So therefore it makes it the same types of bonds that are going to stabilize both of these different types of structures can be found. These components that form these bonds can be found in different types of all different types of proteins. And ultimately, if other interactions help stabilize these conformations, then essentially they can arise and be stabilized as part of the final three-dimensional shape. Proteins can also have what are called domains. Domains are segments of a polypeptide chain that fold up independently of other parts of the protein chain. And usually these segments will have a specific function. So for instance, if we have a chain here and we have this segment here could essentially fold on its own without these pieces. That would be defined as a domain. Here's some examples of different domains of different proteins. And you can see that this particular protein here has 3 different domains. This domain, actually 4, one more here. So there's actually 4 different domains that are found in this mammalian phospholipase protein. So some proteins can have one domain or not, and these domains, these sequences that allow for folding of this proper domain, can sometimes be conserved, and we'll see these different repeated domains in different homologs between different organisms as well. Now, tertiary structure of a protein is when a protein has additional folding that allows it to reach its final three-dimensional shape. This folding, the stabilization of this additional folding, is also due to non-covalent interactions, and usually non-covalent interactions between R groups of the polypeptide chain. And you can see these different types of non-covalent interactions: hydrogen bonds, ionic bonds, van der Waals forces, etc. This happens to show you, this particular figure shows you the tertiary shape, the final three-dimensional shape of ribonuclease, and its specific diffraction pattern by X-ray crystallography, which is a method that's often used to determine the structure of protein, was used to ultimately generate that predicted structure. You can see in this structure there's a helical structure in a few places, there's some sheets, so ultimately they contain different secondary motifs in the final predicted three-dimensional shape of this protein. Some proteins and cells also exhibit quaternary structure, and quaternary structure is ultimately where multiple monomers, single proteins, interact together in a larger protein complex. So hemoglobin is a classic example of quaternary structure. There's 4 different polypeptides that ultimately interact together to create a tetramer, 4 subunits, creating then a larger functional complex, which is involved in carrying oxygen. This is another example of another protein that exhibits quaternary structure. It happens to be a dimer, so 2 monomers that interact together, and then something even larger can also exist like pyruvate dehydrogenase, and it actually has 60 polypeptide chains that form in this larger complex and essentially has quaternary structure as well. Keep in mind that when we think about protein structure, even though proteins fold, these flexible chains fold into their final 3-dimensional shape, and they'll reach their energetically lowest shape due to the maximum amount of interactions that protein can form, that when proteins bind to ligands or other types of proteins, oftentimes we can get subtle changes in the shape, and that's what's called conformational changes. This particular diagram shows you that. It's reverting from an open state to a closed state, and the protein happens to be enzyme acetylcholinesterase, and you can see this is showing you its open state versus that opening will close when it's in a closed state. You can see that most of the protein still remains structurally similar except for subtle differences in shape or active binding sites. So those are different conformational states. And so a lot of times you'll read different papers about proteins that when they bind a certain ligand or bind to another protein, they'll have a conformational state change, ultimately influencing slightly the structure of the protein overall. Myoglobin is another example of a protein in its final 3-dimensional shapes. It was the first globular protein in which its tertiary structure was determined, and it was determined by a method called X-ray crystallography. So it just gives you an idea of this final 3-dimensional shape. Myoglobin is like hemoglobin. It has a heme group that allows it to carry oxygen. It just happens to be a protein that's normally stored in the muscle cells to help increase oxygen levels for muscle when oxygen supplies are getting lower by the circulatory system due to exercise, etc., and it helps allow for increased oxygen levels in a muscle cell. And this shows you 2 different ways in which we can model the structure of myoglobin. Now, the folding process, there's been 2 different alternate pathways that have been proposed regarding how proteins fold. And protein folding is a very popular field in biochemistry that ultimately is studied by different scientists trying to determine the steps of protein folding and how a protein folds, what particular motifs fold first, what looping occurs next to reach its final native structure. And what you can see is there's 2 different kind of examples of how proteins may possibly fold. One of which, the first, is where the protein is unfolded and will first form secondary elements, and then we'll get more additional looping and folding to form that final 3-dimensional shape. And the second model shows you that instead maybe the protein polypeptide chain does some additional looping first, and then the secondary elements will form after that point to reach its final native state. Both are ultimately being explored as far as theories. Protein folding can be studied by doing what's called denaturation and renaturation studies. Proteins in their native shape can be unfolded using certain reagents called urea or other types of reagents. And the protein can then be refolded by removing urea or another type of agent. When a protein is unfolded, it's called denaturation, and when it's allowed to refold again, it is called renaturation. Denaturation, renaturation studies were important because it allowed scientists to realize that proteins have usually one final 3-dimensional shape, because if they unfolded it and then refolded it, it would oftentimes take on the same shape that it did from the start. Now, protein folding is extremely important to function. And in fact, when proteins misfold, it oftentimes can lead to disease states. And there's two important neuronal diseases that involve misfolding of proteins. One of which is Creutzfeldt-Jakob disease, or what we kind of know as mad cow disease, and another is Alzheimer's. This particular slide is showing you Creutzfeldt-Jakob disease, also abbreviated CJD, and there's a protein called prions that are thought to be responsible for this disease. The normal protein PRPC will not misfold improperly. However, there's another version of this prion, PRPSC, which seems to be identical or similar to the wild type, that becomes misfolded. And when it is misfolded, it will lead to degenerative diseases of the neurons. This is found in cows, which is called mad cow disease, or scabies, which I believe is in sheep. But essentially, this type of disease has been found in different types of animals, and they now know it's related to misfolding of prion proteins. This particular diagram is showing you what the normal protein looks like, and this next figure is showing you what the abnormal protein in its unfolded state, improper folded state, looks like, or predicted shape. Another example of protein misfolding having deadly consequences is in Alzheimer's disease. And there's a protein called APP, which is amyloid precursor protein, which can be cleaved into different pieces. And the cleavage pieces, one of them is A beta 42. If there's an increased level of cleavage of this protein, and specifically if there's different mutational changes to APP that increases its cleavage, then essentially you can have aggregates form of this cleavage, and which will help form these plaques, which are a hallmark of individuals with Alzheimer's. And you can see this particular section of a brain of an individual with Alzheimer's and these plaques that are formed. Once again, a disease state caused by misfolding, aggregation of proteins that essentially can lead to a disease state, a very detrimental disease state. Now, when proteins were first starting to be discovered and protein function, there was this study that was done in the 60s that looked at what would happen if we took larvae, Drosophila larvae, and heated them up. And when they heated them up, they noticed that there was an increase in production of a number of proteins. A number of these proteins are now known as what are called chaperones. And at the time they were called heat shock proteins because heat shock meant that there was an increase in these different types of proteins when the cells were heat shocked. Since then, there's been a number of different cell types that have also had heat shock proteins being identified. And we now know these heat shock proteins, most of which are called molecular chaperones. And these chaperone proteins, one of their major functions is to help ensure proper folding of proteins in cells. So most proteins actually require assistance of chaperones to fold properly. Some can self-fold, but others will require the assistance of chaperones. So if you heat up cells, the reason why you're getting an increased level of these proteins is because when you heat up cells, proteins like to unfold and form aggregates with other proteins. And the cell responds to that by increasing these chaperone proteins to ultimately help unfold the aggregates and help fold the proteins properly that have become unfolded in the heat response. After studying these different types of chaperone proteins, heat shock proteins, they found that there's basically two main types, what are called HSP70 and also another category which are called chaperonins or HSP60 family. HSP70 usually bind to polypeptide chains as they're being produced at hydrophobic sites and help prevent other proteins from trying to aggregate with this unfolded protein. And as the protein folds, these HSP70 proteins come off. Larger polypeptides are often shuffled to a larger chaperone complex called chaperonins, and these chaperonins have a cavity in which the polypeptides can move into, and a special environment so the proteins can fold within that cavity property, and then they're ejected. There was this experimental model done more recently after different analysis of the structure of a particular chaperonin protein. And chaperonin proteins, HSP60s, one example is what's called the GRO-ES and EL chaperonin that's been found in E. coli. Now, GRO-EL and also GRO-ES, which is a component of GRO-EL, was synthesized in E. coli, is synthesized in E. coli, and it's important for proper folding of many different proteins. And there's different homologs that have basically been discovered in different cell types as well that have similar structure to GRO-EL and GRO-ES. Two different structural analyses of this complex. This complex is extremely large and made up of multiple subunits. And these multiple subunits form these donut-like arrangements. One of which is there's two cavities on either end of this structure that allows for proteins to fold in. GRO-ES binds to GRO-EL, which you can see in this diagram, and it's a thin cap that's thought to enclose the cavity so that a protein can form. So you can see here's the cavity. So GRO-EL is the larger complex, and GRO-ES is thought to be a cap. Two different structural studies help enable scientists to understand the structure of this complex. One of which was electron microscopy, which is shown here in these images, with and without the cap. So GRO-EL with and without the cap. And also X-ray crystallography studies of the protein itself has also been explored. The more recent X-ray crystallography studies have given even more information regarding the function of GRO-EL and how it works to ultimately allow for proper assembly of larger proteins. The first figure here is showing you GRO-EL without the cap, GRO-ES. When GRO-ES is added to the GRO-EL, to enclose the chamber, what they see is there's a shifting of some of the domains, the protein segments that are on the interior of the cavity. And this particular red domain actually moves out, making the cavity larger. So you can see the shift in this domain here from this diagram when the cap is added. This is thought to be important because when the cap is added, the cavity gets larger. And you can see that also in the previous figure. When the cap is added, it gets larger, which would be important if a protein needed to fold, that the cavity expanded so it would have room to fold into its final three-dimensional shape. From these different studies, they predicted the model, which is shown here. A polypeptide chain would enter the cavity. And it enters because it ultimately will bind to the interior of the cavity. And it binds through hydrophobic amino acids that align the interior of the cavity of GRO-EL. The cap will then be added. And this changes the cavity size. Remember, it will become expanded due to movement of the interior, which will also move the hydrophobic amino acids away and push them inward into the protein structure so they no longer can interact with the polypeptide chain. This will then release the polypeptide chain into the cavity. The protein will then be able to fold into its final three-dimensional shape without any interference of other proteins. And then the cap will come off and the protein will be released. If it's misfolded, then it can re-enter again and try it over again or re-enter another chaperoning, such as the GRO-EL. You can see that there's two cavities that alternate between picking up, assisting in folding, and release, and that this whole process takes about 15 seconds for binding, cap formation, enlargement of the cavity, protein folds, and then protein release. All of which this predicted model was predicted due to different structural analysis by electron microscopy as well as X-ray crystallography that really enabled scientists to provide a model to how this particular chaperone actually works. Now, when we think about proteins in general, also, it's important to note that proteins are lumped together into families, and that's specifically due to the fact that oftentimes proteins have similar sequences. So there's certain conservation of amino acid sequences, and oftentimes these proteins, because they have similar sequence, they also have similar function. These proteins are thought to have arose due to gene duplication earlier on in simpler organisms and different mutational changes to the gene sequences of now these duplicated genes can ultimately lead to subtly different sequences of proteins. And then we get slightly different structures, which leads to slightly different functions, but they may have been once before from the, produced from a single gene in a more primitive organism. Now, another growing field when we study proteins is what's called proteomics. And proteomics basically studies what's called the proteome, which is all the proteins of an organism. And it's a more advanced study, and essentially what scientists do is they collect all the proteins from a particular part of an organism, and they can then run them on gels to separate them out to a certain migrated point using gel electrophoresis, and then they can essentially remove each one of those proteins at a time and study the actual sequence of that protein, which is shown here. So here's the gel which was used to actually separate a complex mixture of proteins, and then these proteins can be singly excised from the gel, digested with peptidases into pieces, and then subjected to mass spec, which will help reveal specific sequences, and then a computer model will basically be able to compare that to theoretical proteins based on gene sequences, and to help determine what gene that protein might have come from. Proteomics is important because we know that a cell's proteins help leads to function of that cell, and an organism's proteins obviously lead to proper function of that organism. So learning about individual proteins and their individual functions helps give us an idea, obviously, about how important those proteins are and what roles they play in cells and in an organism as a whole. And then last but not least, this is the last section of the lecture, and this looks at different methods used to determine protein structure. Some of which you might see, those of you that are presenting next week, used in your papers that you will be presenting. Some of these methods might have been used to determine protein structure of the proteins you're looking at. So one of the first types of methods to talk about is what's called X-ray crystallography. And X-ray crystallography is a method in which proteins first have to be purified, and then the proteins have to be given certain types of conditions, salt concentration, buffer conditions, that force the proteins to form a crystal. And this crystal has to be what's called a perfect crystal. All the proteins have to be aligned perfectly with one another. Once that true crystal is achieved, then you can subject that crystal to a narrow beam of X-rays, and it will hit the crystal and then create a diffraction pattern. The specific diffraction pattern will then be interpreted by computer softwares, and essentially allow a predicted structure to be determined from that diffraction pattern. This is an extremely useful tool to study protein structure as well as nucleic acids. Another important technique is NMR, nuclear magnetic resonance spectroscopy. And this is a little different. Proteins still have to be purified, but you do not have to crystallize the protein to use NMR, which makes it a little simpler. However, NMR can only be used on smaller size proteins. It's like about approximately 40,000 Daltons or less. So you can't use extremely large proteins and try to determine the structure using NMR. You could break it into its domains and study individual parts of the proteins, but you wouldn't be able to study a large protein. And how NMR works is you subject the pure protein to different frequencies of radio waves. That will then produce a pattern that can then be interpreted into positions of hydrogen ions within the structure, which will allow you to provide a predicted three-dimensional shape using computer programs as well. Now, electron microscopy is also used commonly, which is what we saw in the GROW-EL, GROW-ES scenario. It was one of the techniques that was used, as well as X-ray crystallography. But electron microscopy is also used. This is showing you cryo-electron microscopy, which is where they actually freeze the sample and then essentially image it by electron microscopy. This is just going through a step from one lab that uses cryo-EM to study protein structure, basically forming protein in this first step, and then freezing the sample, and then using the electron microscope to look for structures. And this is showing you kind of the images that were produced by electron micrographs. So different, and the proteins will land on a film or a grid, and essentially in different arrangements, and they can look at the different images that are produced to look at, are you looking at the top of the protein, the side of the protein, and then it will give them more of a three-dimensional view of the structure of a protein, which you can see here. And then they can produce different models based on what they can see from these electron micrograph images. Once different techniques can be used, oftentimes they can put them together, such as this drawing. An electron micrograph image is then used in combination with an X-ray crystallography image, and they can position the proteins in such a way to put them together to give them a greater view of a larger macromolecule or protein structure. So combining it really allows for additional information about protein structure, maybe function, which we saw with the GROW-EL, GROW-ES as well. Both of them combined provided useful information to really help to elucidate the function of that Chaperone complex. So this completes our lecture for protein structure, different methods for determining protein structure. And just to remind you, look through the chapter in sections that are relevant to this so you can get a better understanding than even what we've covered here, as well as I have a supplemental reading material on an antifreeze protein in insects and some questions that I want you to focus on when you're reading through the paper as well. So that is also listed on Blackboard. All right, I'll see you guys all next week.